Let's go ahead and get started. Good morning. Am I on? Good. Good morning, everybody. It's nice to see such a nice turnout at 8 o'clock in the morning. That was very difficult for me to get out of bed this morning. As an introduction, my name is Rob McCallop. I'm a new faculty here. I'm a transplant from the state of Virginia. And I have to tell you that I was born and raised a Hokie fan. But due to a lot of pressures through faculty and neighbors, and now even my 5-year-old daughter, I'm becoming a bit of a Gamecock fan. So. My number one allegiance is still the Hokies, but number two, Gamecocks. Shown here is my phone number. If you want to reach me by phone, it's probably pretty difficult. I don't have one of those blinking lights letting me know that there's a message, so I hardly ever check it. More than likely, the best way to get a hold of me would be through my email address. Okay. Today I'm going to pick up where Dr. Mayer left off, and actually we're going to change gears a little bit. What he discussed with you was a little bit about the innate immune response, and then he went into some of the soluble factors that are involved in both the innate as well as the acquired immune response. And what I want to talk to you about is remember the cell-to-cell interactions, a lot of the cellular interactions that are necessary for the development of the acquired immune response. And this first lecture will kind of serve as an overview or an introduction to a number of these different processes, as well as a introduction to a number of the different molecules that are involved in these interactions. And what you will see is that, with the exception of the T-independent antigens, almost all of the responses in the acquired immune response require some type of cell-to-cell interaction. So the first couple slides, I'm just going to give you an introduction to what we're going to talk about in the first lecture. First of all, as I mentioned, this is going to serve as an overview of the subsequent lectures. So in this first lecture, it's going to be a very general lecture, introducing you to some of the concepts. And the following 4 lectures will discuss some more of these aspects in more detail. What you'll notice is that there are a number of different cells that are involved in the acquired immune response, and we'll talk a little bit about each one of those. We'll discuss a little bit about the innate versus the acquired immune response, some of the differences between the two. We'll also discuss the importance of the site occupied by the pathogen. What you will learn is that the site in which the pathogen occupies, either it being intracellular or extracellular, that plays a very important role in determining what type of immune response is elicited. So again, we'll talk about what elicits an antibody response and what is important for eliciting the cell-mediated immune response. We'll discuss a little bit about the different populations of T cells and what role they play in the immune response. And then we'll talk about some factors that determine the specificity of the immune response. We'll talk about the diversity of the different receptors, how you get to the diversity, what is important in the development of the diversity. And I'll introduce to you in this lecture the major histocompatibility complex, but in the next lecture we'll really discuss that in detail. And in the final part, we'll talk about lymphocyte recirculation and migration and how that plays an important role in the immune response. So I guess the first question I would ask you is, what is so important about the acquired immune response? And why is it important that we have both a humoral aspect, which is mediated primarily by the antibodies, as well as the cell-mediated immune response? And the quick answer to that is that we are, as humans, as humans, are exposed to a number of different types of pathogens. And these pathogens vary greatly, not only in the size or their composition, but also in the ways that they infect the cells of our body. And if you want to very broadly break them up into 4 classes, which is shown here, you have the extracellular bacteria, parasites, and fungus. And shown here are some examples of these. Some common ones are the Streptococcus pneumoniae, which causes pneumonia. Clostridium tetani, which causes tetanus. There are also intracellular bacteria and parasites. Some common ones are Mycobacterium leprae, which causes leprosy. There are a number of different viruses that can infect your body. Some common ones are influenza, also HIV. And there are also parasitic worms. Shown here a couple of examples. What's important from this slide and the subsequent slide showing some examples of what these actually look like. Shown here is the parasitic worm that causes swimmer's disease. It results in very bad gastrointestinal problems. We have a nematode here, which causes sleeping sickness. And what you can see here is that this pathogen resides outside the cell. Shown here is a red blood cell. And shown here is the nematode. Example here of a fungus that infects your body. Here's another fungus which causes ringworm. And here are two viruses, HIV and influenza. Now the point I want to make here is not that you need to remember what these pathogens are or what disease they cause. But the point here is that we're exposed to a number of different types of pathogens. And these pathogens, again, vary greatly in the composition, and they also vary greatly in the site in which they try to infect. And basically, you have extracellular pathogens, you have intracellular pathogens. And the immune response to these different pathogens has to be very unique, and they vary greatly from one another. So our bodies are exposed to a lot of different pathogens. Therefore, we have to come up with a lot of different ways to fight these pathogens. So in order to do that, our bodies have developed a number of different cells of the immune system. One common feature of all these is that they're all derived from the bone marrow. So all of your cells of the immune system start in the bone marrow. So this is important, for example, when you hear people getting bone marrow transplants. The reason they're getting a bone marrow transplant is an attempt to boost or to reestablish their immune system. And many times this is in a patient who has received either radiation therapy or chemotherapy. And these treatments, many times, in addition to fighting the cancer, will also deplete your immune system. What we see is that there are two main lineages of cells that are derived from the bone marrow. We have the lymphoid lineage, and these include T cells, the B cells, and the natural killer cells. The T cells and the B cells play a really important role in the acquired immune response, and the natural killer cells are mostly involved in the innate immune response. In addition to the lymphoid lineage, there are the myeloid lineage, and these are primarily the monocytes, macrophage, dendritic cells, the megakaryocytes, and the granulocytes. And we'll talk a little bit more about the macrophage and dendritic cells. And in particular, we'll talk about their abilities to present antigens to the T cells. So shown here is a cartoon of hematopoiesis, which simply means development of the blood cells. And again, as I mentioned, they all start in the bone marrow from one progenitor cell. So this is a pluripotent progenitor cell, meaning that it can develop into many different types of cells. It then breaks off into two different lineages. You have a common lymphoid progenitor. So this is the beginning of all the lymphoid cells. Then you have a common myeloid progenitor, again, which is the beginning of all the myeloid cells. If we concentrate on the lymphoid cells for a second, what we see is that they then can break off into the three main cells, a B cell, a T cell, and an NK cell. So they can leave the bone marrow into the blood, and then you'll see these three types of cells. They further go on to differentiate into the lymph nodes. You see the B cells, the T cells, and NK cells again, and these eventually become the effector cells after being exposed to antigen or being exposed to some other type of stimulus. The B cells can turn into plasma cells. Again, these are the cells that secrete the antibodies. T cells become activated T cells, and what we'll see here is that there are more than one type. We have T helper cells and we have cytotoxic T cells. And then you can have the activated NK cells, which are commonly called LAC cells, or lymphokine-activated killer cells. Now these cells, especially the B cells and the T cells, cannot reach this stage without being or getting some cooperation from cells of the myeloid lineage. So if we go through this really quickly, again, what we see is that from the common lymphoid progenitor, the cells progress into a granulocyte macrophage progenitor cell. These cells then go on to become a number of different cells. And what we were probably going to key on in the next few lectures are the dendritic cells, shown here, as well as the macrophage, which are shown down here. And what's important about these cells is that they can act as antigen-presenting cells. So they're able to sample what's out in the body, process these antigens, and then present them to the T cells and the B cells, the T cells, which then can activate these cells to become activated. But we'll go over that in more detail in subsequent lectures. What's important here is that you know that all cells of the immune system derive from the bone marrow, and that there are two main lineages, the lymphoid as well as the myeloid lineage. So in the next few slides, we're going to actually go into what some of these cells look like, what are some of the functions of these cells. There are small lymphocytes, which are part of the lymphoid lineage, and these cells are mainly the B cells and the T cells. You have natural killer cells, and these are the cells that can kill cells infected with various viruses. Then the B cells, or the small lymphocytes, can go on to become plasma cells. And again, plasma cells are the cells that produce the antibodies. There are neutrophils, and these act by phagocytosis and killing of the microorganisms. Again, we're not going to talk too much about these, but there are also eosinophils. These act by killing antibody-coated organisms, and they release their granular content. So they're activated by coming in contact with antibody-coated microorganisms or pathogens, and they release their granulocytes and kill the different pathogens. The basophils, their function is largely unknown, so we won't talk too much about those. The dendritic cells are a very important cell in the acquired immune response. These are the primary cell that acts as an antigen-presenting cell. Their function is to basically monitor what your body is being exposed to. So they basically will gobble up antigen and then present that antigen to T cells. And that's how the T cell becomes activated. Mast cells, they act by expulsion of parasites from the body through release of granules. And mast cells play a big role in your allergic responses. And again, we have monocytes. These are the precursors to the macrophage. And we'll discuss more a little bit later about macrophage. But they can play two roles. They can kill cells by phagocytosis, and they can also act as antigen-presenting cells. So they also gobble up a lot of the different organisms that can act to present these antigens to the T cells. We won't talk much about the megakaryocytes or the erythrocytes. They don't play much. They play a very important role in the development of the acquired immune response. And this is just to give you some idea of what your blood looks like, what the makeup of your blood looks like with regard to these different types of immune cells. And what you can notice here is that there's a very wide range of the proportion of these cells. The neutrophils can go from 40 to 75%, monocytes from 2 to 10, and lymphocytes from 2 to 20. And this basically really depends on what type, if your body is undergoing an infection, or it also depends on what type of infection. That will dictate these different ratios. I'm going to switch gears a little bit here. So the first part of this lecture is just kind of to introduce to you some of the different cells that play a role in the immune response, and to let you know that there are a wide range of different cells that can act to fight off the different organisms. And most of these, what we're going to talk about, are the ones involved in the acquired immune response. So what I'm going to show you here, talk a little bit about, is the difference between the nonspecific or the innate versus the specific immune response. So what is unique about the specific or the acquired immune response? So when we're talking about the nonspecific immune response, it can also be referred to as the natural, the native, or the innate. So this is your immune response that's in place regardless of exposure to antigen. The innate lacks discrimination between antigens. So it doesn't really recognize specific antigens, but it recognizes situations. So for example, if a cell of your body loses expression of the MHC class I molecule, natural killer cells can recognize that cell and say this is not a normal cell and get rid of it. It's not necessarily recognizing a specific antigen, it's recognizing a particular situation. Although the antigens don't play a role in the development of the innate immune response, the production of cytokines in the acquired immune response can act to enhance this. And again, one example of this is the lymphocyte-activated killer cells. So once the acquired immune response starts developing cytokines, these cytokines can then act on cells with innate immune response, such as NK cells, and activate the cells so that they have better ability to kill different targets. So if we contrast that to the innate or the specific or the adaptive immune response, this is enhanced or induced by antigen. So without specific antigen, the acquired immune response is not activated. Therefore, it shows fine discrimination. So you can have an antigen that varies by one amino acid, and if you inject one antigen that elicits an immune response, if you come back with a second one that even differs by one amino acid, it may not respond to it. So it's very specific for the type of antigen that it's responding to. And an important part here is that it has memory. So if you're exposed to a particular pathogen, you develop the primary immune response. If you're exposed to that same pathogen, say 6 months or a year later, your immune response will recognize that and develop a very strong immune response. And this is the basis for all your immunizations. An important point here is that although these two immune responses, the innate and the acquired immune response, are separate, they also play a very important role in controlling one another. So activation of the innate immune response, in many cases, will go on to activate the acquired immune response. And once the acquired immune response is activated, cytokines and factors produced by the acquired immune response may lead to enhancement of the innate immune response. But they do actually have a lot of crosstalk with one another. So what we'll see is that the site occupied by the pathogen plays a very, very important role in determining what type of immune response is developed with regard to the acquired immune response. So if you remember back, the acquired immune response basically is composed of two separate arms. There's the humoral immune response, which is primarily controlled by development or secretion of antibodies. And then there's the cell-mediated immune response, which is primarily dictated by cytotoxic T cells or cell-to-cell interactions. And what we'll see is that the extracellular, the pathogens that infect the extracellular portion, which is the site of most bacteria, will lead to the development of the humoral immune response. And the reason that is that the antibodies are very capable of interacting with bacteria because They're secreted by the plasma cells into the media, and then they're able to interact with the bacteria. However, if there is an intracellular pathogen, the antibodies can't get into the cell. So the antibody response would not be very effective. So again, the intracellular response is the site of most viruses and some bacteria, and this will lead to the cell-mediated response. And again, it's all about how can the immune system get to these pathogens. Antibodies are not very good at getting into the cells, therefore they would not be the best response. So this is going to be kind of a repeat of some of Dr. Mayer's lecture, I believe. We're going to talk about how these antibodies work. And there are three main ways. You have neutralization. So an example here is that the antibody can neutralize a toxin produced by a bacteria. And the way it does this, it simply binds to the toxin and then basically marks it for engulfment by the different phagocytic cells. You have opsonization. Again, here the antibody will bind to a pathogen. So it's not necessarily just a toxin, it's the whole pathogen. And again, this will basically put up a flag saying this cell needs to be engulfed. And then the third one is complement activation. So what happens here again, the pathogen is covered up by the antibody. The complement then binds to the antibody. The complement cascade is initiated, which leads to the lysis of the bacteria or other pathogens. Shown here is a cartoon of some of these different processes. So if you look at the neutralization process, you have you'll have the toxin, shown here in red, and typically these toxins will bind to receptors on a host cell and then lead to some adverse effect, either killing of the cell or some way enabling the cell. What happens is when we produce antibodies against these toxins, the antibodies bind to the toxins, prevent it from binding to the receptors on the cells. These antibodies then will bind to Fc receptors on phagocytic cells. The toxins will be engulfed and cleared from the body. The second example of an antibody response is opsonization. Again, this is very similar response in that what's happening is antibodies, instead of binding to a toxin, are binding to an extracellular bacteria, shown here. But again, this will mark these bacteria for phagocytosis and clearance from the cell. And the third example is complement activation. This is a little bit more detailed or a little bit more involved, but the concepts are basically the same. You have a bacteria that is recognized by an antibody, and it's coded by, the bacteria is coded by the antibody. The difference here is that these antibodies are able to bind complement. This initiates the complement cascade, which leads to the lysis of the cell. What is the same is that the ultimate result of this activation or of this process is that these bacteria are, again, phagocytized and cleared by phagocytic cells. So the main concept here is that antibodies are able to bind or recognize either toxins or bacteria, and this pretty much labels them for phagocytosis and clearance. And that's shown here. Basically, Fc or complement receptors on the phagocytic cells bind to the pathogenic toxin, complex with the antibody. They become endocytosed and chewed up by the lysosomes. And that's basically shown here. I mean, what you have, again, is a complex, the antibody with a toxin, it's endocytosed, The phagosome then fuses with the lysosome, and then this complex is digested and cleared. Now we're going to talk a little bit about the cell-mediated response. The cell-mediated response primarily is against intracellular pathogens, so it's against pathogens that are able to infect inside your cell and grow inside your cell. Now there are two primary components, intracellular components. The cytosolic, so these are pathogens that basically grow in the cytosol. And this is primarily the site of most viruses and some bacteria. There's also the vesicular. And these are membrane-bound entities. So these are different pathogens that can bind to like your endoplasmic reticulum, the Golgi, or actually into your lysosomes. And this is the site of some bacteria and some other parasites. So again here, what's important is the location will dictate what type of response is elicited. If the pathogen is infecting the cytosol, typically you'll get a CD8 response or a cytotoxic T cell response. So what will happen is the CD8 cell will be activated. It will actually kill that infected cell. So it gets rid of the virus by killing the cell that's infected. However, if it is a vesicular, you can have two different responses. You can have a subpopulation of T cells known as a Th1 that express CD4. They will become activated. They will then initiate the cell-mediated response to clear this pathogen. The T cells also play a role in the humoral immune response. So if the infection is extracellular, another cell population of T cells known as Th2 cells will become activated, and they will then help to activate the B cells to clear pathogen. Is that clear? I think I might be confusing myself up here, so if you have questions, please ask. Okay, so again, I think cartoons are always the best way to try to explain things. I'm always better with pictures. So this is showing the response of a cytotoxic T cell. And again, this is the cell that gets activated following the intracellular or cytosolic infection, for example, a virus. And shown here, what you have is an infected cell. The virus gets into the cell. It then produces different viral proteins or antigens. These viral antigens are then recognized by a specific cytotoxic T cell, which is then able to kill this cell. And this is just an electron micrograph of one such infection. Shown here, the black dots are the viral particles that are expressed on the surface of a particular cell. Shown here is the interaction between a T cell, the T, and the virally infected cell. So this T cell recognizes these viral particles and is now in the process of killing this infected cell. The other way that the cell-mediated response can be initiated is when a macrophage or another antigen-presenting cell is infected with a virus or a bacteria, and it actually engulfs a bacteria or a virus, and it's in its lysosome. This is then processed and presented on the surface. So this is the lysosome. Processes and presents the antigen and expresses this antigen on the surface. A Th1 cell, which is a CD4 cell that helps to initiate the cell-mediated response, will then recognize this particle and produce various factors, typically cytokines. We'll discuss those later, that then activate this macrophage to become able to kill more of these bacteria or pathogens. So the question is, how do these cells, how do the CD8 cells, or how do the CD4 cells recognize these different pathogens? What is so specific about these pathogens, and what is so specific about the T cells and T cell receptors that allows the cell to recognize that a particular cell is infected with a virus? Again, as Dr. Mayer mentioned, the specificity resides in the receptor. So the receptor is the part of the cell that is able to recognize the specific part of the antigen. If we look at the T cell receptor, the one thing that's unique about the receptor is that it recognizes the antigen in association with the MHC complex. And we'll discuss the MHC complex in the next lecture. Another important factor here is that the T cell receptor is univalent. That means it can recognize one epitope. In contrast to the B cells, which recognize antigens and it's bivalent, so it can recognize two. And it doesn't need to recognize the antigen in association with the MHC complex at all. So uniqueness of the T cell is that it recognizes a antigen in context with the MHC molecule and that it's univalent. And each T cell expresses one receptor. Well, it expresses many receptors, but it is specific for one antigen, very similar to the B cell. So now we're going to talk a little bit about how the receptors or the repertoire is developed. And we can go into a little bit about the history here. There were two main hypotheses. One was the instructional hypothesis, and the second one is the clonal selection. Now, the instructional hypothesis states that each T cell, there's basically one receptor. Each cell has one, all the cells have one receptor, and that what happens is that when it encounters an antigen, it's able to mold itself and change itself to recognize that antigen. So this is a very simplistic way of thinking about things. So what they're saying is if your cell's infected with a bacteria, the T cell receptor will see that and then mold itself and change itself to recognize that bacteria. That sounds really great, very simple, but the problem is that doesn't really explain how the body is able to discriminate from self-antigens. So in addition to expressing bacterial antigens, each cell in your body is expressing some type of self-antigen. So if the instructional template was true, what you would see is a lot of autoimmune disorders. So you would expect to see T cells reacting with self-tissue. So the alternate hypothesis was clonal selection. And what this states is that through a number of processes, T cells that are able to recognize self-antigens are deleted, and the ones that are able to recognize non-self are clonally selected. And I'll go into more detail about that. So again, the instructional hypothesis, although it's very simple, does not explain the fact that we're able to recognize self from non-self. So there are four basic principles to the clonal selection theory. First, each lymphocyte bears a single type of receptor of unique specificity. So each T cell is able to recognize one MHC complex antigen combination. And the second part is that the interaction between the foreign molecule and the MHC, the foreign molecule and the lymphocyte receptor leads to a molecule with high affinity, and that leads to lymphocyte activation. So you have to have a very strong interaction between the T cell receptor and the antigen. That leads to activation. The third part is that once you have activation of the cell, all the progeny from that, or of that particular cell, will have the exact same receptor. So if you have a T cell that binds to a particular antigen, it becomes activated and divides, each one of the cells resulting from that division will bear the same receptor. And the important part here is that any lymphocyte bearing a T cell receptor that recognizes a self-antigen is deleted somewhere along the line. So you don't have T cells in your body, or you shouldn't have T cells in your body that recognize self-antigens. Unfortunately, that's not always the case because there are a number of autoimmune disorders that develop, and that's typically because we have some cells that can recognize self-antigens. Shown here again is a cartoon of the clonal selection theory. So again, as we discussed, all the cells of the immune system are developed from one cell. They then go on to differentiate into different types of cells. The first step is to remove any potentially self-reactive cells. So any cell that can recognize self-antigen will be deleted. So that effectively diminishes the pool of T cells. And in fact, it's estimated over 99% of cells that begin as T cells don't make it because they're deleted. Then you have a pool of mature cells, and the one that can recognize the foreign antigen become activated and divide. And each one of these cells that results can recognize the same antigen. They don't recognize different antigens. They recognize the exact same antigen that they were first exposed to. And this is a very generalized cartoon on how diversity is developed. Because in your body, you have T cells that can recognize almost infinite different antigens. So how is that diversity generated? And we'll go into this into more detail in a couple of lectures, but here's a very generalized cartoon. So each cell has a set of inherited genes. It goes through a unique combination. So I think with the B cells, you discussed the B-DJ recombination. Very similar process happens here with the T cell receptors. So you have rearrangement. So one member of each set is joined, so 1V, 1D, 1J, or with the beta chain, with the T cell receptor, it's a V and a J, but we'll discuss that later. And you got one gene, one set of genes that is produced for each cell. So then these then recombine and produce the unique lymphocyte. What's important here is that each lymphocyte only expresses one T cell receptor, specific for one antigen. And it's been estimated that in your body, there are up to 10 to the 8th different types of T cell receptors that can recognize 10 to the 8th different antigens. So in the next lecture, we'll discuss in really great detail the MHC class 1 molecule. But for this lecture, what's important to discuss is what role does it play in the interaction with the T cell receptor. So MHC class 1 molecules are expressed on the surface of all nucleated cells. So almost all the cells in your body express MHC class 1. And this allows the immune system to recognize self from non-self. MHC class 1 is recognized by the T cell receptor on cytotoxic T cells. Again, these are the cells that are able to directly lyse cells. We'll discuss later that CD8, which is expressed by all cytotoxic T cells, it also plays an important role in the interaction between the CTL and the cell expressing the class 1 MHC. Again, this is kind of a repeat, but the source of the peptide expressed with the MHC molecule is always cytosolic. So it's derived from the cytosol. So again, if a virus infects the cell, that viral particle will be expressed with MHC class 1. So again, here's a cartoon of the MHC, is shown here, peptide bound to the MHC molecule, and this is the T cell receptor on a cytotoxic T cell. And this is just a very generalized cartoon of what happens. You have the MHC molecule here expressing the peptide. T cell will come along, using its T cell receptor, will recognize both the MHC molecule and the antigen in combination. Once it's able to recognize one of these, it becomes activated and can kill the cell. Now, in addition to MHC class 1, we have MHC class 2 molecules. These are expressed by far fewer cells. So some nucleated cells, primarily antigen-presenting cells, express MHC class 2. This is recognized by the T cell receptor on helper T cells. And as opposed to CD8 on the cytotoxic T cells, T helper cells have CD4, which helps in the binding of the T cell receptor to the MHC class 2 molecule. And again, the source of the peptide is usually from vesicular compartments. And a lot of these bacteria or pathogens may initiate or originate in the extracellular portion of the body. So what happens is pathogens may be residing in the extracellular portion. They're engulfed by the APCs and then presented with the MHC class 2 molecule. So shown here are a couple of examples. Both the Th1 cell here and this one are CD4 positive T cells. And you have two different examples here. You can have the Th1 cell recognizing a bacterial peptide. So again, what happened here is the bacteria was probably in the extracellular portion, was engulfed by this macrophage and presented to the T helper 1 cell. You can also have a what is known as a helper T cell. And these are usually involved in activating B cells. Sure. Here, typically a class 1 results from an intracellular infection. So if you have a virus infecting the cell, the viral particles will then be processed by that cell and presented. The important point here is, again, class 1 MHC is expressed by basically all cells. All nucleated cells in your body express class 1 MHC. Only a small portion of your cells, and these are primarily antigen-presenting cells, express class 2. Okay, in the last couple of minutes, we'll discuss lymphocyte recirculation. And what we discussed so far is that your body has a number of ways of recognizing different types of pathogens, and it can develop a number of different responses to these pathogens. But an important point is, how does your body how does the immune system come into contact with these different pathogens? If they're just sitting still in one spot, the chances of them actually coming into contact with these different pathogens is very low. So what happens is you have cells of your immune system are constantly circulating throughout your body, picking up different pathogens, sampling what your body is being infected with, and then coming back and basically reporting it to the T cells and to the B cells. That's a very generalized way of looking at it. So you have your secondary lymphoid organs, and these are comprised of lymph nodes and the spleen. And these are the main areas where the lymphocytes encounter the antigen. And as I discussed, you have T cells that can recognize up to 10 to the 8th different antigens. So the amount of T cells that can recognize each antigen is very low. 1 in, say, 10,000, 1 in 50,000 cells can recognize a particular antigen. So it's very low. So again, if these cells weren't moving around, sampling what was going on in the body, the chances of that T cell becoming activated were very low. So it's very important these cells are constantly moving throughout your body. Again, recirculation optimizes the ability to encounter these antigens. And shown here is a very brief cartoon. Shown here would be a site of infection. What typically happens is you have cells throughout your skin. One example is a cell known as a Langerhans cell. It's a specialized dendritic cell that can sample different antigens in your skin. So if you have an infection that starts, say, here at the foot, this cell will pick up antigens from that infection. It will migrate back to a secondary lymphoid organ, such as a lymph node. And you have your T cells and your B cells are constantly circulating throughout your body. So eventually the T cell or B cell that recognizes that antigen come into contact with that dendritic cell. and become activated. And then it eventually, by receiving signals, it will actually migrate down to the site of infection. And that's pretty much summarized here. You have primary lymphoid organs, which are your bone marrow and your thymus. And these are the organs where the lymphocytes are generated. When it comes to circulating throughout the body, your T cells and your B cells go throughout the secondary lymphoid organs. And two of the primary ones are the spleen and the lymph nodes. And this is where they encounter the antigens. And as I discussed before, the APCs, or antigen-presenting cells, pick up the antigens at the site of infection, bring it back to the secondary lymphoid organ, present it to the T cell or the B cell. That then activates those cells and tells them there's an infection down here or at your foot or wherever it might be. And then those cells go down there and help to clear that pathogen. And this is basically summarized here. I think that's it. Any questions?